Dr. Baliga here. I got another terrific podcast today. It's in the realm of evolutionary biology. Unveiling the Hackfish, a gateway to understanding vertebrate evolution. Welcome to another episode of Dr. Baliga's Got Knowledge Doc podcast. Today, we are diving into a groundbreaking study that is shedding new light on the history of vertebrate genome duplications thanks to a remarkable jawless fish, the hagfish. This podcast is derived from a recent paper in Nature titled The Hagfish Genome and the Evolution of Vertebrates. The lead author is Ferdinand Marletaz and the senior author is Daniel S. Rockshar. Both are corresponding authors. There's a third corresponding author, Dr. Jeremiah Smith. The lead author is from University College London, and it's published in Nature Journal 2024, volume 627, page 811 to 820. It's an open access article. The significance of whole genome duplications, how genome doublings shape evolutionary innovation. For decades, evolutionary biologists have been intrigued by how genome duplications shaped the early history of vertebrates. Whole genome duplications are rare, but significant events when an organism's entire set of chromosomes is duplicated, providing fresh genetic material that can drive evolutionary innovation. Susumu Ono, a Japanese-American scientist, generated the 1R to 2R hypothesis. It was a bold prediction on vertebrate ancestry. In 1970, the Japanese-American geneticist Susumu Ono proposed a bold idea. He suggested that our vertebrate ancestors went through not one but two rounds of whole genome duplications, which he called 1R and 2R. Over the years, molecular data and genome studies have supported this hypothesis. The importance of hagfishes and lampreys, the earliest clues to our evolutionary past. Among the earliest diverging vertebrates are hagfishes and lampreys. These eel-shaped, jawless creatures with teeth hold crucial values to our evolutionary past, but the exact sequence of genome duplications in these jawless and jawed vertebrates have been a tough nut to crack. Previous studies hinted that lampreys had an extra genome triplication, but whether hagfishes share this trait was still a mystery. Cracking the code. Understanding when these ancient whole genome duplications happened and whether they were common to all vertebrates or specific to certain groups is key to piecing together the puzzle of vertebrate evolution. Research methodology involved innovative approaches to sequencing the hagfish genome. To tackle these questions, this group of researchers sequenced the genome of the brown hagfish and compared it with the genomes of the lamprey and several jawed vertebrates including humans, mice, chickens, frogs, the spotted gar and the bamboo shark. Their goal was to trace the evolutionary history of genes across these species and infer the history of whole genome duplications. Traditional evolutionary methods have struggled with this because these duplications happened so long ago that the molecular signals needed to reconstruct the events have faded. But the researchers used two innovative approaches to enhance these signals. They inferred duplication from a large number of gene trees and reconstructed phylogenies for entire chromosomes. This allowed them to resolve the sequence and timing of these ancient duplications and triplications with remarkable clarity. What were their findings? The first whole genome duplication shared by all existing vertebrates happened around 530 million years ago, known as 1RV. The jawed vertebrates then experienced a second duplication around 505 million years ago, termed 2RJV. Meanwhile, jawless fishes underwent a separate whole genome triplication over 500 million years ago, called 2RCY. These events clustered around the Cambrian explosion, a period of rapid animal diversification, suggesting that the genome duplications may have contributed to the evolutionary success of vertebrates. Why does this matter? Well, understanding the history of genome duplications gives us a framework to explore how these events relate to the emergence of key vertebrate traits. For instance, many genes involved in forming the neural crest, a group of cells that develop into crucial structures like the cranial skeleton and sensory ganglia, are duplicated copies from the first RV event. This implies that neural crest evolution might predate the first whole genome duplication, a fascinating idea that opens up new avenues for research. Programmed DNA elimination, a fascinating evolutionary strategy. Another intriguing discovery from this study is the phenomenon of programmed DNA elimination in hagfishes. This is where certain chromosomes are present only in germline cells and are eliminated from other cells during development. Similar processes have been observed in other vertebrates like lampreys and songbirds, highlighting a potential evolutionary strategy worth further investigation. The expert opinion and the reviewer of this article said the robustness of the study's methodology, the quality of the genome assembly, and the analysis of synteny and ancestral chromosomes all contribute to a high confidence in these findings. The high-quality genome data has enabled researchers to pinpoint the timing of vertebrate genome duplications with unprecedented accuracy. From Japan to breakthroughs, the journey of hagfish genome research. Behind the scenes, some key moments of this project took place in Japan, where foundational work on vertebrate and hagfish cytogenetics was established. A memorable incident during the study involved a hagfish escaping its tank, a reminder of the extraordinary biology of these creatures. The lead researcher, Ferdinand Marleta's interest in hagfish was first sparked in 2005 during a lecture by French paleontologist Philippe Unveiling, a moment that set a stage for this groundbreaking study. Evolution Unplugged, journeying through the history of vertebrate genomes. This wraps up today's episode of Dr. Balaga's Got Knowledge Doc podcast. Thanks for joining us on this journey through the ancient history of vertebrate genome duplications. Stay tuned for more insights into this fascinating world of evolutionary biology. Until next time, keep exploring.